Dear students, in previous chapter we have learnt about the genes, about the factors, but still the genetic material, that biochemical which is responsible to carry genetic characters or the traits from one generation to the another generation was still unknown. In this chapter, as the topic itself expresses molecular basis of inheritance, we will learn that which biochemical component is responsible for the transfer of genetic inheritable characters from one generation to the another generation. In class 11th, we have learnt about the biomolecule, and we are also aware that there are two types of biomolecule, macro and micro biomolecules. In macro biomolecules, we have learnt about four types of macro biomolecule, that is proteins, carbohydrates, fats or lipids, and nucleic acid. In this topic, we will learn about that out of these macromolecules, which molecule reacts as a genetic material. As you know, nucleic acids are the biomolecule, and these molecules are responsible to transfer inheritable trait from one parental generation to filial generation. That's why these are the molecules, means the nucleic acids, they are the molecules, biochemicals which are responsible for the transfer of inheritable traits. In this chapter, we will study the following subtopics. This is the introduction, what I discussed with you. After that, DNA, structure of polynucleotide chain, and we know polynucleotides, they are the nucleic acids. The means we will study about the DNA of structure of DNA, then replication of DNA. After that, central dogma of molecular biology, DNA packaging, transforming principle, biochemical characteristics of transforming principles, Hershey and cheese experiment. After that, proof of semi-conservative replication of DNA through, that is, Mason and Stoll experiment, process of replication of DNA, how the DNA will take place. Now the transcription we will study under two headings, that is transcription in prokaryotes and transcription in eukaryotes. Then genetic code, then mutation and genetic code. Now mutations are of three types, that is point mutation, frame shift mutation, and silent mutation. After that, we will study about the tRNA, which is known as the adapter molecule. Then translation is the process of formation of proteins from mRNA. Now regulation of gene expression, and we will understand regulation of gene expression with the help of the leg operant model in E. coli. After that, we will learn about the Human Genome Project or HGP, and then last, DNA fingerprinting. So now this is the introductory part of this topic, and in the second video I will discuss with you, that is the DNA is structure.